action potentials. Okay, pretty big on this one, and I'm going to start with the graph and then we're going to annotate all around it. So let's get cracking. So you may have seen the video on resting potential. So we know that when a neuron is at rest, there is a difference in the amount of charge across the membrane caused by the amounts of positive ions on the outside compared to the inside. So we say at rest, the resting potential is, the numbers may vary slightly textbook to textbook. I'm going to say minus 70, and the axis here is going to be the membrane potential in millivolts. You don't need to know these numbers. It's a good idea to be familiar with them to some extent though. Let's start off with, so at rest, we've already ascertained that the resting potential is minus 70. I'm going to put something on this graph which you don't normally find on these graphs, and that's the generator potential. So let's say this is a weak stimulus. It's not reached the magical line, which we're going to call threshold. Okay, so it's not reached threshold. The sodium-potassium pump has returned it to resting potential. Okay, maybe we will do these as we go. First section here, let's do these in black so they stand out. Number 1. Here we have resting potential. What can we say about this? Well, there are a certain number of things they want you to say at all times. And we're going to say here the sodium-potassium pump is what's causing the resting potential to be maintained. What else do we need to mention? Well, it's active transport. And whenever you say it's active transport, you're going to say using ATP. Commonly, you're going to say against the concentration gradient as well. It's not a mark that they put in here. What happens? Well, we have sodium, and from now on, I'm going to refer to sodium as sodium ions, Na+, are diffused out of the axon, and potassium ions, K+, are going to be pushed in by the sodium-potassium pump, and then we can finally say that some of the potassium diffuses out through the potassium ion channel. You're going to see a pattern, not so much with resting potential, but for the other answers along here, at the stages, I'm going to use this as my model answer each time. So the first thing they want to know is the name of the ion channel. It's the first thing they want to know. Second thing they want to know, does it open or does it close? And the third thing they want to know is the movement of ions, which ions move and in which direction. So we're going to apply this 1, 2, 3 to basically all the remaining stages of the action potential. The additional part that's on this graph that's not on most is our generator potential here. You are required to know what the generator potential is, which is why I put it on this graph. What do we need to say about it? Well, keeping these 3 things in mind, generator potential is actually going to be a weak stimulus. What else can we say about generator potential? We can say some of the receptor sodium ion channels will open, but not all of them. So some sodium will diffuse in, but it does not reach threshold. And then we can say the sodium-potassium pump restores the resting potential. Okay, let's continue on this graph. We go back to resting potential for a bit. There's another stimulus. We have a slow increase, and then we hit this dotted red line, which is threshold. I've not actually labeled this axis. This is time, most likely in seconds, or in fact, no, probably milliseconds. This is actually very fast. I don't know why I'm thinking seconds. Threshold. We can say the generator potential reaches threshold. We can say many voltage-gated sodium ion channels. So voltage-gated means they are opened or closed. The gate is controlled by the voltage. So that tells us the name of the ion channel and what happens to them. They're going to open. Number 2 is covered as well. And then we can say what happens to the ions. Well, sodium ions diffuse in. Let's put into the axon. And it's a positive feedback. So sodium ion channels open, and therefore the membrane potential changes, and therefore the more the membrane potential changes, the more voltage-gated sodium ion channels open. So this we can describe as a positive feedback. It causes a very rapid change, is what positive feedbacks do, and we get this rapid depolarization, because the membrane is polarized here, and it's losing its polarity as we go along here. So this upward phase, number 4, there's not very much to say about this. This is depolarization. You can say sodium ion channels are open. In fact, I'm not going to put open, I'm going to say are open. And we can say that sodium is diffusing in. That's why the membrane potential is changing. This is a bit of a combination here. So when threshold is reached is when most of the voltage-gated sodium ion channels open, and the depolarization is the continuation of that. Now we need to repolarize. So it's depolarized. 0 is about here on this graph. This could be plus 35 or plus 40 is maybe what I'm going to write, millivolts. Again, you're not going to be asked. Usually around about minus 55 is what threshold is. You're not required to know specific numbers. We're going to repolarize. And this is going to be 5. What's causing the repolarization? Well, we can say, the mark schemes never say it, but these are also voltage gated. The voltage has changed and therefore they open, so they are voltage gated. We can say potassium ion channels open. They are voltage gated, but you don't tend to get marks for that, and they don't tend to mention it in the textbooks either. I'm going to put it in there so that it helps you to understand why they're suddenly opening now. What happens to the potassium? Well, the potassium diffuses out. If you remember, during resting potential, the electrochemical gradient, we've got loads of sodium on the outside. The potassium channels are open, but not all the potassium can diffuse out because the outside has got so much charge. So there's a bit of a reserve of potassium ions inside the axon that we keep so that we can repolarize it later on. We can also say that the voltage-gated sodium ion channels close. Okay, so we've returned to our resting potential, but what actually happens is we get this overshoot. We get this more than normal polarization, which is going to be number 6, and number 6 is called hyperpolarization. Well, basically, this is when the membrane potential is more negative than the resting potential. And why does it happen? Well, we can say that the potassium ion channels are slow to close, so there's a bit of an overshoot. And then we go back to resting potential, which is obviously explained over here. Now there's one more thing that we need to add to this graph, and that is between the peak of depolarization and the re-establishment of resting potential, we have this period here, and this period here is called the refractory period. I'll keep it in green. So what is the refractory period? Well, it's during this time an action potential cannot be triggered. We cannot start a new one, basically. It also ensures that action potentials are, in the mark scheme or the exam paper, it's going to say discrete. Discrete means not that it's subtle, but they're separate. They don't overlap. So it makes action potentials First of all, we can say discrete. I'll put don't overlap in brackets. And we can also say it makes it unidirectional. Uni is one, directional is obviously with a direction, so this means one way. Because the action potential cannot be triggered during this time, it means that you have to complete one cycle. You can't get halfway through and then go back up again. This graph can't go all down here and then start again and then start again. That doesn't happen. We need an individual action potential, and the all or nothing law is going to tell us that each individual action potential is exactly the same size. If plus 40 is where it happens to be on this graph, then all action potentials will peak out at plus 40. And there's no such thing as a big action potential or a small action potential. You either have an action potential or you don't. And the refractory period means that we get nice clean signals. We get discrete signals, so that you have one and you have another one. And the difference between a light tap of a stimulus and a firm bang of a stimulus isn't the size of the action potential, but the frequency of action potentials. And that's what our brain interprets as a strong stimulus or a weak stimulus.